Hello, I'm going to be talking about synapsis of chromosomes. So we're given this individual with these two genotypes, and now I've drawn the loop and I'll show you how we get these resulting genotypes. So first you've got GHI centromere J KLM. That's this one here. And it's also one that the individual has. You've got GHI centromere J KHG, GHKJ centromere I LM, which is actually the same as this one because they're just flipped. And then MLKJ centromere I LM. So we can look at these and see which ones are viable, because the ones that are viable will have a centromere, no duplications, and no deletions. So we see here that this one's viable. This one is not because it has a duplication of the G. This one is viable, and this one is not because there's a duplication of the M. So it has two viable genotypes in this individual. In a second example, we're given an individual with these two genotypes, which, as you can see, they're the same genotype just flipped backwards, and they are the same thing as this genotype here, which is viable. So both of these will be viable, and since individual 1 and individual 2 both had two viable genotypes, there won't be any difference in the fitness between these two individuals. Here are my references. I used the genetics lecture videos, the PlayPosit videos, for all of my information.